All right, thanks so much for coming to our webinar today. I'm Lyndon Wyatt, Akoya's technical application scientist in the Northeast US, and I'm here with Michael Mclean, one of our scientists in our reagents R&D group. Today, we're going to take a sneak peek at our simulated H&E stain, something that's currently in development in R&D, which will ultimately let us run an H&E stain on the same tissue section that we run our multiplex immunofluorescence assays, so that you can get beautiful morphological information from exactly the same tissue section as you get information on multiple biomarkers. So before we get going, I just want to give a little bit of background on why we do what we do. At Akoya, we're all about understanding the immune interactions that occur in various disease states. Everyone is aware that immune interactions are very complex, but part of the difficulty in understanding them is that the tools that are typically used to probe these complex multidimensional interactions are very one-dimensional and limited, like the IHC stains that we see here. Despite being the gold standard, there are a lot of problems with using monoplex IHC, such as the subjective nature of test results, the overall qualitative nature of these techniques, low dynamic range, questionable reproducibility, and the time-consuming nature of these assays, given the limited amounts of data that they produce. All of these caveats inherent in IHC leave us to ask, what information are we missing when we rely solely on IHC? So we have newer techniques like flow cytometry and sequencing, and these might be great at telling us about the existence of different cell populations and even their relative sizes, or creating cell expression profiles and profiling gene expression. But these techniques don't preserve any of the spatial data that's so valuable, and they can even produce biased data due to the artificial selection inherent in the tissue dissociation process. So these techniques are great complements to IHC, but they can't fully replace IHC because they don't tell us anything about the spatial relationships between different cell populations. Now, over the past several years, we've come to realize how important the spatial relationships between various cells in the immune environment are. First, people looked simply at cell counts or cell densities, or even at ratios between different cell types. Now we find that there's a trove of meaningful information that can be derived by looking at the locations of cells. The rate of publications using our platform specifically for this kind of work is increasing, and we see increasing validation of multiplex immunofluorescence as a technique, as well as validation of our tools over anyone else's to carry out this technique. So back to our IHC stain here, what are we missing out on? When we move into the realm of multiplex immunofluorescence, we realize some enormous advantages. Among other things, we can see multiple biomarkers in the same tissue section, so we don't have to deal with the uncertainty of looking at serial sections. We can save tissue, we can take advantage of the higher dynamic range and more quantitative nature of fluorescence. So this is what we do at Akoya. We make tools for people to investigate multiple biomarkers in single tissue sections, and we do it because the kind of data that you get from these experiments is unique and powerful. Just to give a short overview of Akoya, the company, we have two main product lines, Codex and Phenoptics. And each product line and instrument fits in a different place along the translational pathway that you can see here. Our Codex instrument enables you to look at up to 43 markers in a single tissue section with true single cell resolution, and it's a great fit for discovery and exploratory work. Our Phenoptics instruments, like the Vector Polaris on the right here, they enable you to look at up to 8 markers in a single tissue section and have high throughput. So they're great for advancing biomarkers that you've seen as relevant when running discovery work into a more translational workflow. Today, I'm going to focus on our Phenoptics instruments, namely the Vector Polaris seen here, which was the platform used by our R&D group to develop the simulated H&E stain that Michael is going to talk about later. So our goal with the Phenoptics instruments is to produce highly quantitative data from up to 8 markers as quickly as possible. So how can we image up to these 9 floors at the same time? Our multispectral imaging strategy is what makes the Polaris different and more robust than other fluorescent slide scanners. Typically, when people do this kind of work, the strategy is to use floors with emission spectra that are spaced far apart, and then to use narrowband emission filters to try to separate the fluorescence from each marker. So this technique is easy to do and can work reasonably well for 3 or 4 markers, but even with a pretty low number of markers, you still run into issues with crosstalk. So that's what's shown here. Crosstalk or bleed through happens when you have signal from one marker showing up in a different filter from the filter that it should show up in. So an example here, if we look in the Tritzy filter, we can see the Tritzy filter is capturing mostly fluorescence from the Tritzy floor, but it's also capturing fluorescence from Fitzy and Dappy. So this leads to more signal being attributed to a given marker than should be. Additionally, when you're using fluorescence imaging, you always have some contribution from autofluorescence, which is fluorescence signal that comes from the tissue in general, not from any particular marker. And this varies based on tissue type and fixation procedure. And of course, when we try to expand to more than 4 floors, like the 9 floors shown here, we run into a lot of crowding and crosstalk. So crosstalk and autofluorescence essentially always happen. Looking at lower numbers of markers can decrease the contribution of crosstalk, but it can't eliminate it completely. So what we need is a more sophisticated strategy to deal with these issues. As you can see here, we could try to play that narrowband filter game again, but it would never work because of all the crosstalk we get. On this slide, you can see the results of spectral unmixing. So you can see there's a huge difference between the raw image in the upper left compared with the unmixed image in the bottom left here. You can see in all these other panels how clean each image looks. You don't have any crosstalk from any other markers. For instance, in this cytokeratin image, the image that you see is really just cytokeratin alone. You don't see any contribution from any other markers. Same thing if we look at CD8. You just see CD8 signal and nothing else. You can see this autofluorescence channel at the top. So all of this signal was removed from the other channels, resulting in large increases in signal to noise ratio and much cleaner images. And just to focus on autofluorescence, look at how much autofluorescence we see in this raw image. So you can see here's the raw image, and all of this signal is contribution purely from autofluorescence, which adds like all this haziness in the raw image on the left. So unmixing pulls all of this autofluorescence signal out and isolates it in its own channel. So you wind up with this really nice, clean, unmixed image. Using spectral unmixing as a strategy to deal with autofluorescence is a great thing to do because unmixing is flexible and it's able to handle the wide array of autofluorescence spectra that you see in different tissue types. For instance, here you can see that pancreas and brain tissue, they both have a ton of autofluorescence, but the channel that exhibits autofluorescence most strongly changes from tissue to tissue. So a one-size-fits-all approach to autofluorescence removal won't really work, but the flexibility that you get from spectral unmixing will work really well. Now, when we do image analysis, usually one of the first measurements we look at are simply phenotype counts, the numbers of a given cell phenotype in our image. In this example, we can see percentages of different phenotypes in our image before and after unmixing, and after unmixing. And you can see just how much of a difference there is between the two pie charts. So we see 30 to 50% error in raw images compared to unmixed images. So this just further demonstrates the power of spectral unmixing and how if you're not using unmixing, you can't really trust your images if you're looking at more than 4 markers or so. So by using our approach, we can accurately image 9 colors in a single tissue section, and we can robustly quantify phenotypes and marker expression because of our ability to remove autofluorescence. And what we're showing here is just a 9-color image. So cytokeratin, CD8, FOXP3, CD20, CD68, PD1, PDL1, and KS67. So with our approach, like I said, we can reliably image 9 colors and remove autofluorescence. Now I'm going to give a brief overview of our workflow, starting with our Opal fluorophores. Opal is designed to let you make the most of your tissue by maintaining tissue quality, giving you the flexibility to use different antigen retrieval conditions, enable you to identify multiple markers on the same cell or even in the same cell compartment, and simply let you see more. So like I said before, Opal allows you to look at up to 9 colors on a single tissue section, and it gives you the flexibility to kind of tailor your assay to the work that you're doing, all while retaining spatial context. Our Opal offering is pretty wide, so everyone can really find something that works for them, whether it's through our 7-color PD1, PDL1 panel kits, which are ready to use and come with imaging and image analysis algorithms, or with our detection kits for use on the Leica Bond RX. We have 4 or 7-color kits for the auto stainer. And we also have compatibility with the Ventana Discovery Ultra. One thing to note is that these kits don't come with primary antibodies. Opal uses tyramide chemistry, and this means that because of the way that Opal works, you have the ability to use whatever primary antibodies work well in IHC without having to worry about species cross-reactivity like you do when you use indirect labeling. And I should also add that we have kits for manual staining as well, and we also sell everything a la carte. So again, we have products aimed at any level user from like ready to use out of the box to completely custom. And one other thing I want to point out is that Opal works well with ACD's RNAscope assay. And we have a number of publications using Opal with RNAscope imaged on our instruments for various levels of plexing with RNA fish, and also with mixed RNA probes and protein. Next, I'm going to talk briefly about our instruments. We offer a few different imaging instruments with various levels of automation and image resolution, starting with our Mantra workstation, which does imaging of single slides, and it's completely manual, to our fully automated Vectra Polaris system, which is great for high throughput applications and whole slide scanning, and again, which is what we used for our simulated H&E stain. So just to talk about that for a second, the Polaris is a really amazing instrument. It can do up to 9-color imaging. It has a fast workflow for 7-color imaging, which can produce whole slide images, whole slide unmixed images in about 10 minutes. It can scan bright field and fluorescence at up to 40x magnification, and it has a lot of other nice features like robust slide handling and automatic tissue and focus finding. So just to highlight that fast whole slide workflow on the Polaris, we call this workflow Motif. And as I said before, it enables the user to generate 7-color whole slide unmixed images in about 10 minutes. We output images in a broadly compatible file format, and you can see in this example what this tonsil tissue looks like right when it comes off of the scanner. So this is the raw image. You can see, for instance, all this autofluorescence on the left side here that kind of shows up in yellow. And then you can just unmix this image, and all of a sudden all that autofluorescence gets pulled out, and you can see all the colors are more vibrant. You don't have that haze, and this is all spectrally separated. So you can break this image down into individual layers where each layer corresponds to each marker, and you could do this all on the fly. So again, that workflow is called Motif. This is the same example just with a magnified image, and again you can see, for instance, the blood vessel on the left here. And after we do our whole slide unmixing, the signal autofluorescent signal from that blood vessel pretty much disappears, and the rest of the signals become much more vibrant. We have another workflow, which is a field-based workflow, and this workflow allows the user more reagent flexibility, and it allows the user to do up to 9-color imaging. So essentially with this workflow, you do a whole slide scan, then you annotate the whole slide image, and then acquire multispectral fields of these stamped areas. So like I said before, this can take your experiments to 9 colors, and you can again output images at 10, 20, and 40x in a widely compatible image file format. And this last slide just highlights the flexibility of the Vectra Polaris. We really see it as a solution for any digital pathology need. It does amazing fluorescence imaging of up to 9 colors with autofluorescence removal. It can do RNA-ish. It has a fast whole slide multispectral imaging workflow. It performs really well as a bright field slide scanner, where it can actually do bright field unmixing too for multiplex chromogenic assays, or even for DAB or H&E. And it also has a really nice TMA workflow available for bright field and fluorescence, which really streamlines the TMA imaging process. Last, I'm going to talk about software. So Inform is our image analysis software, and it has 5 main functions. The first function is unmixing, and that's what we're looking at here. So what we see here is a raw image right when it comes off of the Polaris. This image shows just the contribution from autofluorescence. So this is after unmixing. All of this autofluorescent signal is isolated and pulled out of the image. And then this is the unmixed image. One other nice feature that Inform has is called pathology views. Pathology views allows the user to render these immunofluorescence images as pseudo IHC images. So we see a pseudo DAB image here, It can also just do pseudo hematoxylin if we want to look at the nuclear signal alone. The next main function that Inform performs is tissue segmentation. So tissue segmentation uses machine learning and allows the user to quickly and accurately segment images. Next is cell segmentation, which allows you to segment cells into nuclei, cytoplasm, and membrane. And this is really important so that you can get information from your analysis on the location of different markers with subcellular resolution. Then phenotyping. Phenotyping again uses machine learning to assign user-defined phenotypes to cells quickly and accurately. And then scoring, where you can score cells, where you can score cells based on expression using several different scoring functions, including H-score, which is shown here. We also have the ability to do a few different types of spatial visualizations and produce spatial analyses, like the nearest neighbor analysis shown here, and like the touching cells analysis shown here. And finally, well, while we're talking about software, finally, I want to talk about Proxima, which is the newest addition to our software portfolio. So Proxima is designed to solve two main issues, issues related to data storage and image management, and issues related to the need for faster image analysis and portability between software platforms. I'm not going to go into too much detail here, but Proxima solves these issues by taking advantage of cloud computing. Just at a high level, Proxima provides an image management solution with unlimited cloud storage and the ability to do rapid whole slide image analysis. Along with this comes nice features like collaboration tools, a safety net for file deletion, a secure storage environment, and a seamless way to transfer images to third-party image analysis software. So with that, I'd like to hand it over to Michael Mclean, who's going to discuss the simulated H&E stain that our R&D group's been working on.
Thanks, Lyndon, for that overview of our technology. And I think that really sets the stage and provides some context for my portion of our presentation, which is on the simulated H&E, in quotes there, for MIF treated sections. I'm Michael Mclean. I'm an associate reagent scientist with the reagents R&D group here at Akoya Biosciences. So let's briefly go over what is the traditional H&E stain. This is a bright field stain using hematoxylin and eosin that highlights acidophilic and basophilic elements of the tissue sample in pink versus blue, purple, depending on your bluing steps potentially, respectively, with eosin being pink and hematoxylin being the blue or reddish purple stains. It is necessary for pathologist review. Textbooks are laden with H&E images, and it elucidates tissue morphology and can reveal disease states to people who are trained to look for these things. It'll only allow for indirect comparison of sister MIF to H&E stained tissue sections, and this could be at a distance of perhaps one serial section up to maybe even dozens away. And cancer samples in particular can change pretty dramatically depending on how far away sections are from each other. It cannot be removed such that fluorescent IHC or MIF could be performed on the same section. You'll end up with, if you're trying to use reasonable removal methods such as ER, you'll end up with hematoxylin remaining still visible to the naked eye. Eosin will hang around as sort of a haze in the greenish yellow emission spectrum. And a portion of all tissue sections must be dedicated only to H&E, obviously. And depending on your study requirements, that could be one tissue section or a greater number of tissue sections dedicated only to this particular stain. So what are the Akoya SH&E reagents? This is a fluorescent basophilic and acidophilic dye combination. It's not quite a modified H&E stain so much as a simulated H&E stain, but it uses very similar chemistry to mimic the staining patterns and intensities very accurately of the traditional bright field hematoxylin and eosin stain. It does not use autofluorescence as its eosin mimic. These are truly dyes that stain the same things that hematoxylin and eosin will stain. It's performed after rehydration, prior to antigen retrieval and MIF. Your monochrome images from this stain can be colorized to represent the traditional stain, and chromatic elements like melanin or what appear to be oxidized RBCs can be incorporated in your simulated H&E images via a bright field overlay, as I'll demonstrate in the following slides. So this allows for multiplexing. I've put it in quotes since it's not technically existing on the same section at the same time, but it is in fact on the same section, on the same tissue sections, and direct comparison of your H&E images to your MIF images. So I'll give you a window into some images from the development of this tool. On the top left, you can see a colon cancer sample that has been stained with a true hematoxylin and eosin stain. This is a bright field slide scan acquired on a vector Polaris microscope. And to its right is a sister section from the same colon cancer sample stained with the SH&E fluorescent dyes. And this has been unmixed and colorized to represent the traditional hematoxylin and eosin stain. And you'll notice from left to right, there are some minor chromatic details that don't necessarily resolve under fluorescent imaging. These images may be good enough under certain circumstances, but if you would like for those things to be incorporated, it is possible via what is represented along the bottom, that being on the far left, a 20x magnification frame from the sample immediately above it, showing some RBCs in dark red there, appear to be RBCs, flanking some colon versus cancer tissue cell types. To the right is, again, a 20x mag frame from the sample, again, immediately above it. This is the fluorescent SH&E stain that has been colorized to represent traditional H&E. And to that image's right is the true bright field image, actually, from that fluorescent stained sample. And then that has been overlaid to the right. So these are in fact images taken from the same sample at the same region and then overlaid to represent the traditional bright field H&E. So this slide contains the 2020 SITC poster introducing this technology. I will move very briefly through this. There's a link provided along the bottom of the screen here. You can also find this under the literature tab on our website at akoyabio.com. I'm going to go over a few individual figures from this with you. This is figure 1 from the poster. You can find it sort of front and center there. It takes up maybe about a fifth of the page on the poster. I think the images perhaps speak louder than words for this specific tech. You can see the traditional H&E. This is a lung cancer sample, a human lung cancer sample. The traditional H&E stained version of this is, again, on the left-hand side. That's a full slide, well, a full tissue slide scan, again, acquired on a vector Polaris microscope. Along the top, along the bottom is a 20x mag frame from the same sample. The traditional H&E, represented in blue and pink, is displayed on the left. To its immediate right is sort of a pseudo-colored fluorescent image of a sister section of this same sample, with the Akoya SH&E basophilic stain being represented in cyan and the acidophilic stain being represented in magenta there. And you can sort of adjust the intensities of these things as you would be able to with other fluorescent dyes, retrospectively or retroactively. And then this scan, the monochrome images from this scan have then been just converted into sort of a bright field format and colorized to mimic the traditional H&E appearance to the right-hand side there. So that's actually, those are different versions of essentially the same image from the same scan of this tissue sample. And then following this SH&E staining and scanning, this tissue sample has been subjected to heat-induced epitope retrieval, which is sufficient for removal of this stain. So it's what would ordinarily be included in your workflow anyway, and then subjected to Akoya PD1, PDL1 Motif lung cancer kit staining for those 6 protein targets and DAPI, and that is represented on the right-hand side. So this can give you sort of a direct window of comparison here between your H&E image and then your MIF image. So hopefully this provides a good window into the utility of this tool. And this figure just demonstrates the versatility of this stain. I've provided 3 different human cancer samples stained with traditional H&E. Along the top, you can see a breast cancer sample followed by, again, a colon cancer sample and a melanoma, and then the respective SH&E colorized bright field overlaid images for sister sections from the same samples right along the bottom there. And then a brief overview of the workflow, that being, one, you would bake your sections as usual, rehydrate them potentially through xylene, ethanol, distilled water, and then perform the SH&E stain in a single aqueous application. Contains both of the dyes. Cover slip this image, decover slip, and then perform AR or HIER as you would, and then perform your MIF stain subsequently. So why would you want to use this? Well, it's a highly accurate representation of the traditional true H&E bright field stain performed before AR. A pathologist-friendly H&E image is achieved for every sample, which will have direct relevance to your MIF results from that sample, whereas previously perhaps you would have your H&E sample be far enough apart from an MIF sample that the two really can't be compared all that effectively. It might allow for the replacement of H&E with SH&E, of course, depending on the requirements of your study. So you could be able to dedicate more samples to MIF that would otherwise be dedicated to H&E, true H&E. It's quick, it's easy. Again, one single aqueous application. It's non-hazardous and it's photostable. I've tested scanning these things, I think up to 15 times, and I haven't seen any decrease in emission intensity after that much imaging. And it allows for the storage of samples before MIF. In my hands, I've stored samples under organic medium for up to a month and seen no difference across paraffin embedded versus the SH&E stained and stored samples using the same staining method across the two of them at the same time. And finally, you can triage your samples based on the SH&E annotations and the appearance of these things under what would be an H&E representation for inclusion or exclusion in your study and MIF staining. So with that, I'll wrap up my presentation. Thank you for attending today. And I think we're available now to answer questions.
Okay, great. Thank you so much, Michael and Lyndon, for those excellent presentations. So now we'll answer a few of your questions. We ask that you please use the Q&A function as you submit your questions, and we'll go through and read as many as we can in the time allotted. So the first question I see is, too, at the same time, when is this technology going to be available for purchase? So today, our simulated H&E is still in development. So this is really an R&D sneak peek that we wanted to give you guys as part of Akoya Academy. So right now we're currently planning to run reproducibility studies. But the reason why we wanted to put this in front of you is we're really looking forward to hearing from you, the researchers, about what features you'd like to see and how you'd like to use this solution to further your studies. So please reach out to your local representative, and we're happy to have those sorts of conversations with you. So no release date as of yet. Let's see. So I think this one is for Michael. What process or mounting media do you use to remove the cover slip after imaging the simulated H&E?
Yeah, so I tend to prefer cytosol. So that would be a series of dehydration steps followed by cytosol mounting. And then the process to remove that would simply be to dissolve the cytosol after imaging, to dissolve the cytosol in xylene, and then rehydrate prior to MIF. There are aqueous options as well, but it would simply be to leave the slide in the solvent until the cover slip falls off.
Okay, great. Another question. I think this one is for Lyndon. I'll throw this one over to you. Will running the simulated H&E before multiplex interfere with antibody specificity or co-localization?
Thanks, Victoria. No, running the simulated H&E won't interfere with any antibody specificity downstream. You should still be able to see like 3 to 4 markers co-localized on a given cell. Opals only bind within around 10 nanometers of the target epitope, so we don't have too many markers that have membrane colocalization issues. So yeah, the short answer is no. Running SH&E shouldn't interfere with anything downstream.
Okay, great. Here's one that I will also throw to you, Lyndon. Is there any decrease in tissue quality due to the additional steps of the simulated H&E?
The answer is also no. We don't really see any decrease in tissue quality from the extra step.
Okay, great. Oh, here's a question again for you, Lyndon. How does the Polaris know how to distinguish autofluorescence from true signal?
Oh, okay, that's a great question. So that has a lot to do with how Polaris actually does on mixing, or how our software does on mixing. And I say the short answer is basically that the autofluorescence spectrum is distinct from the spectrum that you get from your floors. So the unmixing software takes these spectra kind of as inputs to do unmixing. So since there's such a difference between the autofluorescence spectrum and the spectra from all of your floors, that's how it's able to do unmixing.
Okay, perfect. So Michael, could you maybe fill in a gap a little bit more on the removal step? How exactly do we remove the simulated H&E prior to multiplexing?
Yeah, I just wanted to quickly chime in on that. It's actually done via AR methods that you would use normally. So there's not necessarily an extra step. It's what you would ordinarily do with the FFPE tissue section prior to MIF staining anyway.
Perfect, thanks. And here's a question kind of along maybe the same vein for you, Michael. So after you've stained with the simulated H&E and now you've done your Opal, how long are the slides generally stable for?
It would be the same as regular Opal, so around 6 months in the dark at 4 degrees Celsius.
Perfect. Okay, so another one for you, Michael. Why not use the autofluorescence channel that the vector Polaris isolates as an eosin mimic?
Good question. The patterns, while you can certainly see tissue in the AF channel, the pattern, the intensities of that they don't quite match what you would expect from the traditional bright field eosin stain. Whereas actually using an acidophilic dye will resolve the pattern that you would otherwise be anticipating.
Perfect. Lyndon, this one is for you, and this is in regards to the RNAscope that you mentioned. How does the Polaris and Inform help with RNAscope analysis?
Sure. Yeah. So the Polaris is a really flexible imaging instrument, and I think one of like the great features of it is it's able to do, regular fluorescence like protein imaging. It can do IHC, bright field IHC, and it can also do fish imaging or chromogenic IHC. One of the reasons that Polaris is so useful for doing fish is because of its ability to do unmixing. Usually when people are doing fish, they're only looking at around 4 colors at a time. So unmixing probably isn't strictly necessary in all cases to separate the signal from the probes, but it's really helpful to have unmixing to separate autofluorescence from any of your signals from your probes. So this is especially helpful when you have a probe in the FITSI channel, because FITSI is really prone to autofluorescence. So Polaris can unmix the autofluorescence from the true signal of the probe so that you can produce, strong and clean signals. The other thing that's really helpful is that Polaris can image at 40x. And this is great for, looking at the small dots that you see when you do ISH. And a lot of people use RNAscope, the RNAscope assay from ACD. For fish, which works really, really well, and ACD actually has a Polaris, and they use this 40x workflow with unmixing after to produce really good results.
Perfect, and we have a few questions on analysis with the simulated H&E, but Lyndon, I'll just distill a few of them. Hopefully, this gets at it. Can you run the simulated H&E and then still bring the results through Inform into a third-party platform like Halo or Visiopharm?
Yep, absolutely. Yeah, you can. Running the H&E before, or running simulated H&E before your multiplex doesn't affect your ability to do downstream analysis. So you can run the simulated H&E and then just do everything like you're doing now. So take it into Halo, QPath, or do analysis in Inform. Nothing changes there.
Perfect. So I think we have time for one last question in the remaining minute. And Michael, this one is for you. How quick is the scanning on the Polaris of the simulated H&E at 40x? Is it any different than regular? And does the scanning capture a single image?
I mean, it's about the same time that you would expect if you were taking a 40x scan with a fluorescent, like a traditional, MIF image. So, I mean, in my hands, it takes maybe around 20 minutes, between 20 and 30 minutes, depending on the size of the tissue section. If you're scanning at 20x, it's maybe around 10 minutes. Again, it's very similar to what you would experience with your MIF tissue sections. The exposure times aren't terribly long.
Perfect. Okay, and the last question is, can I get a link to the recording of this webinar? And the answer to that for everyone is yes. This webinar is being recorded and will be up on our website, on our webinar page in just the next coming day or two. So that today is all of the time that we have. And for all of the questions that we didn't get to, we have them all logged, and we will be reaching out to you guys via your individual representative and make sure that we can answer all the questions that you still have. So I'd like to thank you all today for your time and for attending our webinar. and hope to hear from you and continue working with you guys to put out the best-in-class products. So again, thank you so much and enjoy the rest of your day, all. Bye-bye.